Welcome, an initiative by Kekewa College of Horticulture, Nasik, from Department of Soil Science. I, the Professor Ines Gogre, will going to deliver an audio-visual learning for students on topic.
Proteins. The name protein was suggested first by Swedish chemist John Jacob Berzelius as a complex nitrogen substances found in cells of living beings. What do you mean by protein? Proteins are molecules, that is macromolecules formed by amino acids. Proteins are large size molecules made up of structural units called amino acids. A total 20 of different amino acids exist in proteins, and hundreds to thousands of these amino acids are attached to each other in a long chain to form a protein. In brief, proteins are the polymers chain-like molecules produced by joining number of small units of amino acids. That is typical definition of proteins. Then, then classification of proteins. Based on chemical nature, structure, shape, and solubility, proteins are classified as classification based on composition. It can be classified into simple proteins, conjugated proteins, and derived proteins. So let's study about simple proteins. Simple proteins are defined as the proteins that upon hydrolysis yield only amino acids or their derivatives proteins, as they composed of amino acid residue. On hydrolysis, these yields proteins only constituent amino acid. It is further divided into fibrous protein, example keratin, elastin, and collagen. Then globular proteins, example albumin, globulin, lutein, and histone. Then albumins. These are widely distributed in nature and more abundant in seeds. Soluble in water and dilute solutions of acids, bases, and salts. These are coagulated by heat and usually deficient in glycine. Example, leucocene in cereals, whole albumin from white of egg, then serum albumin from blood plasma. Then globulins. These are of two types as the pseudoglobulins and euglobulins. Pseudoglobulins. Pseudoglobulins of milk whey, then euglobulins, serum globulin from blood plasma, whole albumin from egg white, then gluteins. These have been isolated from only plant seeds. Then protamine. These are basic proteins which occurs in animal of mainly in sperm, strongly basic yield and cheaply basic amino acids upon hydrolysis. Example, clupine from herring sperm, salmine from salmon sperm. Then histones, soluble in water, dilute acids and alkalis, but insoluble in dilute ammonia, readily coagulated by heat. Example, globin of hemoglobin, nucleohistones of nuclei. Then protamines, found only in plant seed, which is insoluble in water and dilute salt solutions. Then solution dilute acids, alkalis in 60 to 80% of alcohol, not coagulated by heat. That is protamines. Example is gliadin from wheat, zein and corn, hodin from oat. Then scleroproteins, commonly occur in animals and hence animal skeleton proteins, which is generally insoluble in water, salt solutions, dilute acids, and alkalis. Example: collagen of bones, elastin in ligaments, keratin hair, keratin in hair and horny tissues. That is all about simple proteins. Then let's study about conjugated proteins, also known as a complex protein or heteroprotein. The conjugated proteins are the proteins are linked with a separable non-protein portions called as the prosthetic groups. The prosthetic group may be either a mental sorry prosthetic group may be either a metal or a compound. On hydrolysis, these proteins gives amino acids as well as a prosthetic group. And conjugated protein is classified into glycoproteins, chromoproteins, lipoproteins, nucleoproteins, and phosphatin. Then let us study about glycoproteins. These proteins contain carbohydrate as a prosthetic group. Glycoprotein contains small amount of carbohydrate, less than 4%, while nucleoproteins contain comparably higher amounts more than 4%. Example of glycoproteins are egg albumin contain certain serum globulins and serum albumins. Then chromoproteins. These are proteins are coupled with colored pigment. Example: hemoglobin, myoglobin, hemoerythrin, and cytochromes. Then metalloproteins. These are proteins linked with various metals such as Fe, cobalt, manganese, zinc, copper, and magnesium. Then phosphoproteins. These are proteins linked with phosphoric acid. Example: casein of milk, vitelline of egg yolk. Then lipoproteins. These are formed by combination of proteins with a lipid such as lecithin, cephalin, and fatty acids. Example: lipovitelline, egg yolk. Then nucleoproteins. These are compounds of simple basic proteins in salt combination with nucleic acids as the prosthetic group. These are most abundant in tissues. Example: nucleoproteins from yeast and thymus and also viruses. That is all about conjugated proteins. Let us study about derived proteins, that is C. These are derivatives of proteins resulting from the action of heat, enzymes, or chemical reagents. These are further grouped as primary derived proteins and secondary derived proteins. Let us study of primary derived proteins. It is classified into proteins, then metaproteins, and coagulated proteins. Let us study about proteins, which is insoluble in water, appear as a first product produced by action of acids, enzymes, or water on proteins. Example: elastin derived from elastin, myosin derived from myosin. Metaproteins. It is insoluble in water but soluble in dilute acids and alkalis. Produced by further acids or alkali on proteins at about 30 to 60 degrees centigrade. Then coagulated proteins, which is insoluble in water, produced by the action of heat or alcohol on proteins, that is coagulated egg white. Then secondary derived proteins. These are derivatives of proteins in which hydrolysis is certainly occurred. The molecules are as rule smaller than the original protein. It is classified into proteoses, peptones and polypeptides. Proteoses soluble in water, coagulated by heat, produced when hydrolysis proceeds beyond the level of metaproteins. The primary proteoses are sorted out by half saturation with ammonium sulfate and precipitated by nitric acid and picric acid. Example: albumins of albulin and globules of globulin. Then peptones soluble in water, non-coagulated by heat, produced by action of dilute acids or enzymes when hydrolysis proceeds beyond proteoses. It is neither salted by ammonium sulfate and nor precipitated by nitric acid or picric acid. Then polypeptides. These are combination of two or more amino acids units. In fact, the proteins are essentially long chain polypeptides. It is all about classification on the basis of composition. Then classification on the basis of structure. It is classified into fibrous proteins and globular proteins. Then let's study about various functions of proteins. Proteins are vital for growth and repair, and their functions are endless. Proteins act as a storage of energy. The milk protein casein stores amino acid. Then proteins are composed of amino acid, serve in many roles in the body, serve as a building block units of cellular and organic structure. Protein are important structural elements of the body. Example: hair, wool contain keratin. Certain proteins, specifically in animals, are involved in the transport of many essential biological factors of various parts of the organism. Example: hemoglobin and lipoproteins. Some proteins help in regulating cellular or physiological activity. Example: insulin regulates sugar metabolism. Proteins are molecular instruments through which genetic information is expressed. Protein also acts as a catalyst, that is enzymes, which can control the rate of biological reactions and physiological functions. Example: urease, amylase, catalase, cytochrome C are enzymatic proteins. Then some proteins are endocells endocells and organisms with ability to contract, to change shape, or to have move about. Example: actin and myosin function in contractile system of skeletal muscle. Some protein acts as toxic substances such as snake venom, bacterial toxins, and toxic plant proteins like ricin. These toxic proteins are also have defensive functions. Many proteins defend organisms against invasion by other species or protect them from injury. The example is Immunoglobulins, that is antibodies, can precipitate or neutralize invading bacteria, viruses, or foreign proteins from another species. Then they perform hereditary transmission by nucleoproteins of the cell nucleus, that is whole albumin, gluten, are storage proteins. That is all about functions of proteins. Thank you.